Welcome to AllAboutRx. Today, we're diving into a biochemistry foundation that's absolutely vital: carbohydrates. Whether you're gearing up for a massive board exam or just brushing up on your foundational chemistry, you are in the exact right place. We're going to break down their structure, classification, and stereochemistry. So here's our game plan for today. We'll cover biochemistry and carbohydrate basics, move into nomenclature and reducing sugars, tackle structural complexity, dive deep into stereoisomerism and optical activity, and finally wrap up with a quick recall to lock it all in. Okay, Section 1, Biochemistry and Carbohydrate Basics. Let's start with a foundational definition. Technically speaking, carbohydrates are polyhydroxy aldehydes or polyhydroxy ketones. And if you look at their empirical formula, CH2O to the nth power, you'll notice something pretty cool. They literally reflect carbon and water in a 2-to-1 hydrogen to oxygen ratio. It's just a brilliant reflection of how they're formed natively in nature, mostly through photosynthesis, where chlorophyll converts carbon dioxide and water into sugar.
But, why do we actually care about this in a medical or clinical setting? Well, carbohydrates aren't just about energy. They are fundamental diagnostic tools. We rely on them to uncover the root causes of genetic disorders, and tracking blood sugar levels is an incredibly powerful primary screening tool. This is exactly why clinicians obsess over biochemistry. Understanding these molecules is the absolute foundation for monitoring diseases like diabetes, dictating rational treatments like a sugar-free diet, and seeing how well a patient is actually responding to therapy.
Moving right along, Section 2, Nomenclature and Reducing Sugars. When we're looking at simple sugars, we generally split them into two structural camps based entirely on their carbonyl group. Try to visually anchor this in your mind right now. If a sugar bears an aldehyde group, like you see highlighted in blue, we call it an aldose. But if it features a ketone group, highlighted in maroon, it's a ketose. That one single structural difference, it dictates a massive amount of the molecule's chemical behavior. Now, what's genuinely interesting here is how beautifully logical carbohydrate naming conventions actually are. We use Greek numeral prefixes to tell us exactly how many carbons are in the chain, and then we just slap the suffix "-ose at the end. So 3 carbons, triose, 5, pentose, and this pairs perfectly with our aldose and ketose rule. Take a 6-carbon sugar, like glucose for example. It's an aldose, so specifically it's an aldohexose. Fructose, also 6 carbons but with a ketone group, is a ketohexose. Once you spot the pattern, you can instantly picture what a sugar molecule looks like just from its name alone. This brings us to a massively important concept for your lab exams: reducing sugars. So what actually makes a sugar reducing? Well, if a sugar has a free aldehyde or ketone group when it's in its open chain form, it has the chemical power to reduce cupric ions, that's Cu2+, down to cuprous ions, or Cu+. This electron transfer is the vital chemical mechanism behind classic diagnostic tools like Benedict's and Felling's tests. If the solution reacts, you've got a reducing sugar for sure. All right, Section 3, Carbohydrate Structural Complexity. Carbohydrates scale up in complexity through things called glycosidic linkages. At the most basic level, we've got monosaccharides, the simplest units, like glucose. Link 2 of those together, you get disaccharides. Chain 3 to 10 units, and you've built an oligosaccharide. When you start linking more than 10 units, sometimes thousands of them, you're looking at polysaccharides. And finally, we have derived carbohydrates. These are specialized sugars that have been altered by oxidation, reduction, or maybe the addition of amino groups. It's literally like biological Lego. Okay, let's dive into a quick study hack. Disaccharides are going to show up on almost every single biochemistry exam, and you need to know exactly what they're made of. To effortlessly memorize the most important ones, just remember this incredibly simple mnemonic: some ladies may. That simple phrase maps perfectly to sucrose, lactose, and maltose, letting you quickly match them to their building blocks. Some is for sucrose, which is glucose plus fructose. Ladies is for lactose, which is galactose plus glucose. And may stands for maltose, which is just 2 glucose molecules linked together. Definitely keep that one in your back pocket for test day.
Now, when those simple sugars form massive homopolysaccharide chains, meaning polymers made of only one type of monosaccharide, they become vital nutritional reserves. In animals, we store our excess glucose as glycogen, which is a highly branched polysaccharide. Plants, on the other hand, store their glucose as starch. Both serve the exact same biological purpose. They act like a densely packed battery of energy just waiting to be broken down when the organism needs fuel.
Let's keep going. Section 4, Stereoisomerism and Optical Activity. This is exactly where exams lay traps, so pay close attention. First up, we have epimers. By definition, epimers are two isomers that differ in configuration around one specific carbon atom, and crucially, this has to be a carbon other than the carbonyl carbon. Think about glucose and galactose. They share the exact same chemical formula, and they're completely identical everywhere in 3D space except at carbon 4. Because of that one single shift, they're C4 epimers. However, if that configurational difference happens specifically at the carbonyl carbon, which remember is C1 for aldoses and C2 for ketoses, they aren't epimers anymore. We classify them as anomers. This structural change is exactly what gives us the alpha and beta types of sugars, like alpha glucose and beta glucose. And this brings us to enantiomers. These are exact, non-superimposable mirror images of each other. It's kind of like your left and right hands. They're mirror images, but you can't stack them perfectly on top of each other. For sugars, whether we designate a molecule as the D isomer or the L isomer is dictated entirely by the green-tagged penultimate carbon. That's the asymmetric carbon that is farthest away from the carbonyl group.
I want to pause here and give you a massive exam tip. Never confuse the anomeric carbon with the penultimate carbon when you're assigning these names. The anomeric carbon is strictly for your alpha and beta anomers, while the penultimate carbon is the sole reference point for your D and L enantiomer designation. Make sure you keep those two totally separate in your mind.
So here's the crucial point, and it's honestly the biggest trap in biochemistry. Do not assume that a D-designated sugar automatically rotates plain polarized light to the right. D and L's designation is strictly about the spatial structure around that penultimate carbon we just talked about. The plus and minus signs, on the other hand, dictate optical rotation, meaning which way the actual substance bends light in a polarimeter. They are absolutely not the same thing. Actually, D-fructose is the perfect example that proves they're completely independent concepts. Structurally, it's categorized as a D sugar, but it's actually levorotatory. It rotates light to the left, which gets a minus sign. Also, keep in mind that a racemic mixture has perfectly equal amounts of plus and minus isomers, which results in zero net optical rotation. And meso compounds? Well, they have asymmetric carbons, but they're internally symmetrical, so the two halves essentially cancel each other out, again giving us no rotation. All right, we made it to Section 5, Quick Recall and Outro. Let's see how all these foundational rules come together. Go ahead and grab a screenshot of this high-contrast summary right now, because these are the absolute core takeaways you need to have memorized for your upcoming exams. Remember the empirical formula. Remember that reducing sugars need a free aldehyde or ketone to reduce copper. Burn "some ladies may" into your brain for your disaccharides. Know the strict differences between epimers, anomers, and enantiomers. And whatever you do, never forget the optical rule. D does not equal plus. Because knowing these visual traps exist is really only half the battle. How will you apply this structural map on test day when you're staring down a massive, complex branched polysaccharide? Think about how these tiny atomic-level structural shifts completely alter biological function. Thanks for tuning into this explainer here on AllAboutRx. Be sure to check the description for the full study notes and keep your pharmacy foundations strong. We'll see you in the next explainer.